name is Tom Miller, and I am the director of the UMC's Chesapeake Biological Lab, and it's my pleasure to welcome you to our Science for Citizens program. UMC's is part of the University of Maryland Center for Environmental Science. UMC's is Maryland's graduate university for the environment. We conduct research on a wide range of environmental issues that help provide information to you, the citizens of Maryland, and to people who make decisions on our behalf in the legislature, in the executive branch, at both state and federal levels. We have been hosting Science for Citizens events at CBL for over a decade now. These events have always been in-person, free, public seminars, and we have amassed about 50 or more of those over the years. And when the coronavirus pandemic hit, we were challenged by how to respond. And after a great deal of thought, we came up with the idea of a virtual science semester, of which Science for Citizens is a part. The other components, which are available on our website, include visits behind the scenes at CBL, parts of the campus that visitors don't usually get to see. They include Science at Home, which targets middle school children. They include videos with our current students, asking them why they chose to come here, what they hope to learn during their time with us, and what their aspirations are for the future. And then interviews with some of our alumni who have been out in the world for 5 to 10 years, and we ask them about what the important lessons were that they have taken from CBL and what role their education has played in their lives. We're able to do this because of the generous donations of 3 sponsors, PNC Bank, Southern Maryland Toyota, and Team Hyundai, who normally pay for tea, coffee, and refreshments at our Science for Citizens lectures, but have generously allowed us to use those funds to reach this broader audience through this virtual science semester. So we thank those sponsors and are delighted to welcome you to what is a very poignant evening for us within UMC's. Today we learned of the loss of a longtime UMC's faculty member, Michael Kemp. Michael started his career as a postdoc here at CBL and moved over to Horn Point when Horn Point was first established in the 1970s, and had been a long-time faculty member at Horn Point throughout his career, and made critical and important contributions to our understanding of the Chesapeake Bay and of estuarine and coastal waters more broadly. Throughout his career, he collaborated closely with Walter Boynton, who's on our faculty here, and many of you have heard of Walter speak in these presentations. Michael was one of the most intellectually and personally generous people I have met in science, and his loss is a real tragedy. It's a particularly poignant night because tonight's speaker is our own Jeremy Testa. Jeremy is an associate professor here at CBL and also studies estuarine and coastal ecology, and in fact received his PhD under Michael's guidance from Horn Point in 2013. When we interviewed for an assistant professor position at that point, Jeremy had not yet finished his PhD. All of the other candidates we interviewed had at least 4 years postdoctoral experience. And it is a testament to the training that Jeremy received, in that although he was the most junior of the candidates we looked at, intellectually he was the broadest and the most mature of all those people. And my colleagues and I have never regretted for one moment the decision to hire Jeremy. He has brought intellectual curiosity, he has brought enthusiasm, and he has brought a highly collaborative spirit to CBL that in itself serves as a testament to his advisor. Tonight, Jeremy is going to talk about events in the Patuxent River and the history of those events and how the history of those events affects current conditions. We operate these evenings as a Zoom conference rather than a Zoom call, so you're unable to unmute your microphone, but that does not mean you cannot ask questions. We encourage you to ask questions through the chat function. And if you click on the chat, we will see your questions. And at the end of this evening's presentation, I will moderate questions for Jeremy, and he will provide answers to those questions. I will try and choose as broad a range of questions as we possibly can. And so without any further ado, it is my distinct pleasure to introduce tonight's speaker, Dr. Jeremy Testa, who's going to be talking about history and the ecology of the Patuxent River. Jeremy, over to you.
Thank you, Tom. Thank you for that really nice introduction, and just following up on some of your comments about Michael, although maybe many of the people who are joining us this evening may not have met Michael. He was a very special person to me, and I actually first started working with him in 2003, and it was on my master's thesis where I was starting to study some of the long-term change that has happened in the Patuxent, and I revisited some of that work, which now seems somewhat dated, putting together the presentation tonight. And I guess would like to follow up on that to express, I think, how this presentation tonight would be something that Michael would very much appreciate because of its attempt, at least, to try to understand how a big ecosystem has changed for a number of different reasons over a long period of time. So I'll be thinking of him as I'm talking to you this evening. So as Tom eloquently put, I'll be talking tonight about change in the Patuxent River and the Patuxent River estuary, and spend a little bit of time talking about its tributary companions. And what I mean by its tributary companions are all the little creeks that flow into the Patuxent, where I've been able to do some particular work since I've come to CBL with my colleagues Walter Boynton and Laura Harris, who collaborate on that project with me. And so really what this presentation tonight is about is a mystery. And although I don't want to overstate the fact that it's a mystery, and perhaps I feel like we shouldn't be in a situation where we have big mysteries when we think about how these ecosystems change over decades, it turns out that in the Patuxent, I think we do have one. And although all great mystery novels end with maybe the resolution of that mystery, I will give you a teaser that we are not going to find such a resolution tonight, but at least in trying to explore what some of these answers to the mystery are, you may get a little bit of a feel for how the Patuxent works and how it's changed over time, and how that has implications for the way we think about all different regions of Chesapeake Bay work. Okay, so one of the reasons that I'm saying that we have a mystery on our hands is because the Patuxent estuary, the river that flows into the estuary, is one of the places in the region that has had pretty well-defined reductions in the amount of nutrients that come into the system. And I think for most of you that have engaged in these presentations before, you have a sense of why we think adding too many nutrients to a coastal water can have problems. But if you look over time at various metrics of what we might call health by looking at a variety of different aspects of the system, whether it be the chemistry, the organisms that are there, and some of the gases that are present in the water. If you look at these types of metrics overall, you can see that for the Chesapeake Bay, which is the green, you can see that there's been a relatively stable score over time. And in fact, if the year 2019 was removed from this, where we had really exceptional rainfall and the pollution that that is associated with, this Chesapeake Bay health score has actually increased somewhat. But if we look at the Patuxent scores over time, they're typically lower than the main stem of the Chesapeake Bay. And if anything, they may have ever so slightly declined over time. And I think when report card grades come out for the Patuxent, they're often in the C's and maybe the D's, right? And I think many of you have noticed that before. And so the mystery is, well, if this is a place where some measurable pollution reductions have been implemented and achieved, why is it that the system seems to be stuck in a state that's opposite to what we would have expected? Okay. So a couple of things that I just want to point out about this system that many of you might be familiar with. On the left here, you can see a map, a couple different maps of the estuary. And there's a lot of little dots and numbers and codes, and what they all represent are different stations where something has been measured over the past 30 years at least. This could be water chemistry, this could be the amount of algae in the water, this could be some estimate of fish. And so we actually have a fair amount of information that's been regularly collected in this ecosystem to try and understand it. Another aspect of this, if you look at the figure that's sort of green, overall bay background with the Patuxent River watershed, which is in the red, you can see that there's a number of orange squares there, and those squares represent the location of wastewater treatment plants. And the Patuxent is somewhat unique compared to some of the other tributaries of the bay in that wastewater treatment inputs are one of the more important parts of the nutrient balance. At least they were. And one of the reasons for that, if you look at this snap that I pulled off of Wikipedia in a Google search, you'll see that red line on the Google map follows the river up into its watershed. And you can see that it's pretty well flanked by the metropolis of Washington, D.C. and the metropolis of Baltimore. So there's a lot of people that live in this watershed, and it's been changing a lot over time. So even when you leave the watershed and enter the estuary, you find a place that changes quite substantially as you move from its sort of freshwater streams as you go down into tidal waters. And so again, we look at this map of the watershed, and if you look in the northern part, which is just at where Route 50 crosses over the Patuxent, you may have noticed, we really have a stream, a freshwater stream that's relatively narrow. If you start moving downstream in and around Jug Bay, kind of in the vicinity of Upper Marlboro, you have this really narrow river that's completely flanked by large tidal marshes that themselves do a lot to change the biology and ecology of the river. And as we get down to Solomon's, where we are, you have a tidal, relatively salty river that has a fair connection to Chesapeake Bay. So the system itself is pretty complex. A lot of the tributaries of the bay are complex. And these changes as you move along the gradient from land to bay, from low salinity to high salinity, the system changes quite substantially. Okay, now even in this watershed, the land that surrounds the estuary has changed quite a bit over time. These are some maps that were put together by a former colleague at CBL, and my colleague Walter Boynton provided me. But what they illustrate is from the middle of the 19th century, what the land use was like in the watershed. And so in 1850, you can see this watershed looks yellow, and it was almost entirely covered in agriculture. As time went by, some people moved west for better farming opportunities. The watershed changed, and you can see the green start coming back after the Second World War, and that indicates that forests were regrowing in those former fields, and you start to see red popping in there, which is the increasing development and growth that occurred in that boom period. As you move into 1972, you can see that actually the forests continue to expand, but the reds get a little bit more abundant. And as you move into the middle of the 90s, you can see that that forest is actually starting to retreat some in favor of suburban development. So this is a watershed that's changed over time, and it's related to these inputs of wastewater that, at least historically, were important in this system. So another way that the watershed has changed is that the human population, as you might have indicated or gathered from the last slide, has increased exponentially over the past few hundred years. And so this is just an example of that. This is the population of Calvert County from 1790 to 1990. And you can see that relatively substantial growth over time. And if you add what at least the state of Maryland thinks will be the population in the 2020 census, you can see that roughly another 20,000 people are expected to be added to the county. And if you add up the total number of people in Calvert, St. Mary's, and Charles County that are expected to be added at the 2020 census, that adds up to about 76,000 people. And so having more people in the landscape is one of those changes that can have large impacts on these estuaries that I think many of you are aware of. Okay, so I want to transition to talking about the ecology of the estuary and how it works and put that into a perspective of time. And to do that, I kind of want to illustrate the most basic conceptual model of at least what the efforts to restore these estuaries are targeting, and what the implication of those efforts are. So when we think of, or traditionally what's been thought of as a healthy ecosystem, is one where there's relatively low nutrient inputs. And so nutrients are important, just like they grow the grass on your lawn when you put fertilizer on them. If you put them in tidal waters, they'll grow the microscopic plants there really well too. And although some of that growth is good because it feeds the food web of the system, too much of it can have consequences. And so in this idealized condition where nutrients are low, you have less of these microscopic plants growing in the water. The water is a bit clearer, so it's allowing more light for the grasses that grow on the bottom. And when those microscopic plants decay, they take up oxygen. And when they're at low levels, that pressure to take up oxygen goes down. So this is kind of the target at its most basic level of what's being sought after. So this just contrasts with the opposite of that, where you have high nutrients, you have high algae growing in the water. When they sink, they decay and take up oxygen, and they can cloud the water, and just the high nutrients themselves can support the growth of these microscopic cells on the leaves of the plants themselves, and they don't do as well. And so there's this contrast, this constant tug between trying to recover the system back to this perhaps previous state of low nutrient inputs. And, you know, at least from the management community's perspective in terms of restoration, these metrics that are referred to, or that are associated with algae and with oxygen and grasses, they all come down to establishing habitat for fish and crustaceans, right? And the idea being that the system becomes more fishable and supportive of, you know, more of the organisms that we like to catch. Okay, so let's talk about this nitrogen input history of the Patuxent. This is another figure that my colleague Walter Boynton shared with me. And what he and some of his former students were able to reconstruct is a time series, at least going back to the 1960s, of what the total nitrogen input to the Patuxent River was. And that's what TN indicates on this graph. And so if you walk through this kind of interesting pattern, this bell pattern over time, in the early 60s, there was apparently, you know, or presumably less nutrients available. But it was also a period that was really dry. It was sort of a drought period for the better part of a decade. And as a result, without having so much precipitation falling and pushing water into the estuary, there was just less overall nutrient input. Now, this was followed by decades of development associated with those population increases, and the nutrient inputs to the system went up relatively rapidly as a result. But in these efforts to start to try to restore the system, bring the nutrients back down to a level that was deemed acceptable, technology began to emerge that allowed for more of the nitrogen and phosphorus that was in wastewater treatment plants to be removed. And so therefore you could have a similar amount of people living in a watershed, but you could really take out the nitrogen associated with those people before it got to the estuarine water. And so B&R is called biological nitrogen removal. It really utilizes a process that happens naturally in these systems and uses it to turn nitrogen from a form that algae really like to one that can degas. And so that started in the 1990s, and there was a relatively, an arguably modest decline in the nutrient loads that resulted from that. It was helped along by a relative drought that happened between 1999 and 2002. But you can see when you have a really wet year, like 2003 that followed, the loads come right back up. But then when that flow resides, they come back down. So there's this complex history of the nutrient loading to the system, and it kind of sets the stage for how this estuary has changed over the past several decades. So this is just to give you an example, and I'm going to go through a couple of slides that in real brief terms kind of describe some of the degradation that happened to the estuary in response to the nutrient loading increases that started in the 70s. So these are plots on the left from stations upstream of the Benedict Bridge. For those of you who know where that is, it kind of starts to delineate where the more fresher part of the system kind of opens up into the saltier part of the system. And then on the right is downstream of that Benedict Bridge. And these are concentrations of nitrate, the most bioavailable, one of the most bioavailable forms of nitrogen in the system, over the course of a season. And so really what I want to communicate here is that from the early 30s through the 60s, and then on after into the 70s and 80s, the nitrogen concentrations more than doubled in the system, right? So it just means that the system's set up to be in this sort of less ideal state. Okay. And so I've presented this in talks before, and maybe others have, and but one of the consequences of having those nutrients increase is that the grasses that grew widely in the system started to disappear. And so these are images from the Chesapeake Biological Laboratory where we are in 1933, and you can see this blanketing of these grasses around the island. And if you move into the 60s, you can see that maybe there's less of those grasses, but they're still fairly dense covering the island. And sometime in the early 1970s, those grasses all but disappeared. And in 1999, you can see that there's really no evidence of that. So a more objective, quantitative look at that is this figure to the right. And note this is a logarithmic scale, so that means this decline is actually a lot more substantial than it actually looks. But this is the aerial coverage of these grasses in the estuary, with data and images patched together for a couple of the historic years. But it really illustrates that the coverage of these plants really went down across the estuary. And I guess the last time I gave a Science for Citizens talk, it was high on the heels of when grasses actually returned to CBL, the shores right in front of CBL, the first time since the early 70s. And that really followed along a period of relatively lower nutrient inputs and perhaps more importantly, a relatively dry period so that there was persistently low inputs. But those grasses have since disappeared with the extreme rain that we had in 2018 and 2019. And so I'm just sort of teasing the idea that, you know, things with the climate are important in how these systems work. Okay, so there was this degradation. And because of the degradation, there were nutrient management actions taken to try to restore the system. And so one of the major actions that was taken in the Patuxent and many other places around the Chesapeake region as a whole was that new technologies were implemented to reduce nitrogen inputs. And so this is just two graphs that illustrate over the past 35 years or so how much nitrogen has come into the lower part of the Patuxent River. Now, the overall numbers are bigger if you look at the whole basin, the part of the watershed that goes far upstream. But this just reflects what happened a bit further downstream, more local to the salty waters of the system. And so all I really want to point out here is that the total phosphorus load went down dramatically right at the beginning of the time series in the early 80s and began a relatively slow decline that continues until this day. Now for nitrogen, there was a relatively high input in the 80s and the 90s. And then when this first treatment process was put in place, the load went down substantially. It actually started to climb up a little bit. And one of the reasons for that I'll come to in a second. And then a second more advanced sewage treatment process was put in place in the mid 2000s. There was another drop and the loads have stayed like that since. And so there has been a relatively stable decline in these inputs. All the while, the total discharge from these plants has been going up. And although these data only go up until about 2003, it just gives you a sense that with more people in the watershed, the overall discharge from these plants is going up, but the load is going down. So the technology is actually really effective in, allowing much more people to live in a place while having, at least for those who are sewered, a fairly stable impact on the system. Okay, so what happened? Was this successful? What did we learn from this? Well, these are maps of trends in the total amount of nitrogen and the total amount of phosphorus that are measured in the surface waters at dozens and dozens of stations across Chesapeake Bay. And if you just zoom in to the Patuxent, where you see these triangles pointing downward, those are decreases that are somewhere between 0 and 50%, right? And that are significant over time. So what I really want to point out here is if you look at the top left of this red box, you see the upper parts of the estuary further up in connection to the watershed have gone down over time. Whereas when you move downstream, there's really no trends that are evident from this process, right? So you have these sort of grayish diamonds, which mean that not much has really changed. So this is an early indication that although there was this relatively substantial decline in the loads, the nutrient concentrations only reveal its effect in these sort of lower salinity waters. If we look at a similar trend map provided by the Chesapeake Bay program and their team, and we look at chlorophyll, which is our sort of simple measure of how much algae are growing in the water, you can see that in that upper, again, low salinity part of the Patuxent, there's significant decreases, this time over the whole past 35 years or so. But as you move into the lower part of the estuary, once you get south of the Benedict Bridge, you get closer to Solomon's, you see that the arrows are going in the other direction, which is the wrong direction. So even though nutrients went down, chlorophyll came up in those lower bay regions. So another thing that happened in the upper bay that seemed positive was that the coverage of these SAV, or these submerged plants that are one of the target restoration measures, that when phosphorus removal began in about 1985, there's really no change in these plants. But once the nitrogen removal kicked in the early 90s, these plants, their coverage really jumped up. And so if you put together the 3 slides that I just showed you, you can kind of start thinking about the story where these wastewater treatment inputs in the upper part of the basin declined. The nutrient concentrations went down, the amount of algae went down, and the amount of plant cover came back up. So it fits this simple conceptual model of how a system might turn itself back around and head towards more of a restoration-like state. Okay, so that's what I just said. But there's a but here, and that's where the mystery comes in. It's that despite this relatively substantial and expensive nutrient load decline, and although there's evidence of a recovery associated with that in limited parts of the low salinity system, as indicated in the chlorophyll maps, the lower saltier region did something different, okay? So now this brings us to some of the way ecologists like to think, theoretically, if not specifically, about how these systems change in response to pollution. And so this is a graph that comes from one of our colleagues that thinks about how these nutrient inputs change the system and how we have this expectation that when we reverse the pollution that's occurred, that we just assume that the system's going to recover back to some state that we remember, which they cheekily refer to as returning to Neverland. And it turns out that returning to Neverland, meaning this red line, if you look in the top left, this red line sort of shows, in this case, the algal concentrations going up with nutrient loads. And then when you reduce those loads, the concentrations come down basically the same way in which they degraded. But the authors kind of illustrate a few other options for that, where we or the system never quite gets back to the place it once was. Or if it does, it takes a different path to get there. So let's just use the regime shift option here on the top right. There's a sort of exponential increase in chlorophyll with more nutrients. And then once you take off the nutrients, the chlorophyll really stays the same. There'd be no evidence that anything was getting better until you reach a certain point, which is fairly low nutrient inputs, and then you have a rapid decline. These are all hypothetical, and we have evidence that different systems work in this way in different circumstances. But I use it to introduce the idea of what's happened in the saltier parts of the Patuxent, because they seem to indicate that we're not going to return to Neverland, and we're not sure when we might get there. Okay, so what's one way that we're not getting to Neverland? So if we look at this, again, a metric of this algae in the estuary over the past 35 years, we see that the chlorophyll concentrations are either stable or going up slightly. And if we look comparably at this measure of Secchi depth, which is one of the simplest tools we use in science, is this disk here on the right that basically measures how transparent the water is. We can see that that depth is going down, meaning the water is getting cloudier. So nutrient inputs decline from these wastewater treatment plants, but chlorophyll is stable or slightly increasing, and the amount of light in the water, at least this measure of it, is going down, okay? The opposite of what we might have expected to occur. These are data where different invertebrates that live in the sediment in the bottom, which are very sensitive to the water conditions, how they've changed over time for sort of a limited period, only going back to 1995. But you can see that the abundance of these organisms and two measures of kind of their diversity have all gone down, which is the opposite direction, right? So again, this is not exactly what we would have expected to occur with a nutrient load reduction. And in fact, if you overlay a measure of how much of the estuary has really low oxygen water, which is indicative of pollution, you can see that this measure has really not changed much over time. And you can see that when those that low oxygen water is at a high volume, the green, the benthic richness goes down. You can see them kind of going in opposite directions from one another, really giving some indication, at least with these data, that the system has not changed, and as a result, some of these biological measures have not improved. And so this is a map of that dissolved oxygen. And this is a map for the entire Chesapeake Bay, put out by the Maryland Department of Resources. And if you sort of zoom in the middle, you can see the Patuxent on this sort of bird's eye view of the bay kind of flipped on its side. And where you see the really bright red on this figure is where there's almost no oxygen in the water. And this is from this past July. And so it's clear that there's been relatively consistent low oxygen conditions that have persisted in the system over time. Okay. So to summarize where I've been so far, these wastewater treatment improvements have happened, but the Patuxent, the saltier parts of the Patuxent, continue to degrade. We have low oxygen volumes that have persisted. Chlorophyll in the broader estuary or algal biomass is sort of the same, and these health metrics remain poor. So the question is, well, what happened? Okay. Well, as I said before, I can't possibly solve that question today, but I want to at least spend the rest of my time laying out some potential reasons why that could be the case. So the first idea is that there could be nutrients coming from somewhere else. There's a watershed in the lower part of the Patuxent that we don't really measure that well that might be contributing nutrients. The Patuxent exchanges openly with the main stem Chesapeake Bay, which is another eutrophic system. Maybe there's inputs there. You know, we kind of typically talk about phytoplankton or algae or these plants that grow in the water as if they're sort of living independently and they only respond to their food, right? But they also get eaten by a lot of organisms in the water. So maybe the food webs have changed in some way that have allowed these algal populations to persist, even though nutrients may have gone down somewhat. You have to wonder if some other forces changed. In Dr. Woodland's seminar last week, you know, he talked about some of the temperature increases that are pretty clear across the region. Is there other forces like that have changed over time that have sort of overrun the nutrient issue? And then there's these more, let's say, theoretical questions about, could a place like the Patuxent kind of be stuck in its degraded space because the system has just changed because it's been polluted and it's just not as resilient as it used to be, or it's accumulated so much pollution that's still available that even though you cut off the source, it's still feeding itself from the bottom. And so I'd like to try to explore all of these in brief. Okay, so let's start with number one, nutrients from somewhere else. Well, this is a graph over time of nutrient inputs from different parts of the system. And the color of the arrow on this map to the right corresponds to the color of the line to the left. So if you look at what's coming from upstream, which reflects some of those wastewater inputs, but also reflects the way the populated watershed really acts, you could convince yourself that those loads have declined somewhat over time. And in fact, the amount of nitrogen delivered for a certain amount of river flow has in fact gone down, right? So there has been somewhat of a reduction there. But if you look at what I call the local inputs, which is our best guess at what's washing off from the sides of the saltier part of the estuary, those loads have stayed constant and they're relatively big, okay? So that there are other sources of nutrients that we may not have really considered as much as we should have. And then if you look at the red line, which is the local wastewater inputs to the downstream estuary, they've gone down, but they're actually not a big part of the budget, right? So even though they've gone down, other sources of nutrients have stayed stable, and the ones that have stayed stable are relatively bigger. Okay, so let's talk about another potential option. Does the main stem Chesapeake influence the lower Patuxent? There's this open exchange, the tides come in and out. Could it be importing nitrogen into the Patuxent? So this gets back to some of the work I did when I was a master's student, working with Michael Kemp, where we were looking at kind of the same type of question, but we only had data till about 2003. And we used this really simple kind of mass balance approach that allows us to calculate how much nitrogen is either leaving the Patuxent and going to the main stem or coming into the Patuxent from the main stem. And where this input typically happens is in the deeper water. And if you look over time, this graph really shows that this increasing line means that there's more and more nitrogen coming into the lower part of the Patuxent from Chesapeake Bay. And another way to think about that is if it's coming in the bottom water, right? It's got to find its way up to the surface at some point, right? And the surface is where it's going to grow algae. And so this graph on the bottom, the bar graph, it really illustrates the ratio of how much nitrogen is coming from the bottom versus how much is coming upstream. And that ratio was appearing to creep up over time, meaning that there's more and more of the nitrogen balance of the lower system coming from the bottom. So this would indicate that maybe it's from the bay, maybe it's from other processes happening in these deeper, channel basins of the Patuxent that are just continuing to produce nutrients. Okay. So that's related to this idea that's actually been fairly pervasive when people have looked at how lakes and how estuaries respond to these managed reductions in nutrients. And the way that people have articulated this is thinking about it as memory. So these estuaries, the Patuxent, for example, the idea is that it's accumulated so much material over time that has nutrients in it, and it's all sunk and accumulated in the sediments, that the sediments are just continuing to cough up nutrients even though the load's declined, right? So this little diagram here shows, an external load coming from the watershed, and then what we could call an internal load coming from the sediment. And the idea is that when you reduce that external load, maybe the internal one is continuing to generate nutrients to keep the water in somewhat of a polluted state. And the reason why something like this might be at play is that residence time, right, which we think of is the time it might take new water to sort of flush out of the system, is, at a median level about 70 days. So water stays around in the Patuxent for quite a long time. So there's lots of opportunity for the plants living in the estuary to take it up. Okay, so there's one last consideration here when we talk about this memory effect. And that's that if you have low oxygen conditions, they tend to change the way nutrients are cycled in such a way that you get more recycling of nutrients, and therefore the system kind of continues to feed itself. And so I just want to illustrate that with this diagram here, right? So you have a really simple cycle of nitrogen that's occurring in an estuary. So you might have this material, this solute called ammonium, one of the labile or one of the sort of munchable forms of nitrogen that's in the system, being taken up by algae, right, to get particulate nitrogen. It sinks to the sediment. And there's a process when there's oxygen around called, it will be degraded into ammonium, and some of that leaks out and can sort of feed the system, keep cycling. So if there's oxygen around, some of this ammonium, NH4, can be changed to a different form of nitrogen called nitrification, and then denitrified, which turns it into a gas, and it's removed from the system. And this is in fact the process that sewage treatment plants take advantage of to take nitrogen out of the system. So if there's oxygen around, there's the capability to kick out some of that nitrogen from the estuary. Now, if you contrast that with low oxygen conditions that persist in the Patuxent, all of these same processes are at play, but there's no oxygen. It creates chemicals that inhibit this transformation of ammonium to something that could be denitrified. So you're not removing nearly as much of nitrogen from the system, and in fact, it just gets recycled and continues to feed the system. And people have estimated that a molecule of nitrogen, at least in Chesapeake Bay, can be used 7 times before it's ultimately buried, degassed, or exported. So these systems are just really good at recycling the nutrients they get. So one of the ways that we've been able to actually put some numbers on this over time and try to understand whether or not this sort of internal effect could be at play is that my colleague Walter Boyan and his colleagues for decades, and us recently on these Patuxent cruises that Dr. Woodland talked about last week, we take intact cores of the sediments and we incubate them, and we can measure how much nutrient is coming out of that sediment and how much therefore would be being recycled back into the water column. So if we look at a couple of these stations where we have really long records, and these are remarkable records to have these kind of measurements over decades. If we look at 2 places, one of them, the red line is from near Benedict, and the blue line is at the mouth of St. Leonard's Creek down by Solomon's. You can see that the oxygen concentrations have in fact seemed to come down over the period of this record. And the amount of ammonium, this labile nitrogen form, have been coming out in greater quantities over time, suggesting that, well, perhaps there is this internal source of nutrients that have been persisting in the system over time. But the record only goes to about 2002 and 2003, so we just don't really know what happened in the interim. But if we kind of take an average of what we've measured on the Chesapeake Bay Patuxent cruises, just in the past couple of years, you might, at stations that are close to these, you get an indication that at least these numbers haven't appeared to come down, and they seem comparable to what was last measured 20 years ago. So this at least gives some indication that maybe there is this possibility to have some nutrient recycling that's kind of keeping the system in the state that it's in. Okay, before I finish, I want to talk about one last possibility, and that's that the food web's changed. Okay, and so as I said before, one of the traditional ways people think about algae is that, and in fact, the way the whole restoration is kind of geared is that the more nutrients you have in the water, the more algae you have, right? But there's something that we call top-down control, and that's that all the organisms that graze and eat those algae can actually control their biomass quite substantially. And so there might be ways that whole changes in the food web of the system could actually affect the amount of algal biomass that we measure. So I just want to articulate one possible pathway that this could happen, where there's nutrients that can feed phytoplankton, right, or algae, but there's zooplankton, right, sort of the insects of the Patuxent, for lack of a better word, that can be grazed by the ctenophores, which are the jelly, sort of the, losing the word for it, it's too late in the evening, but the sort of the bulbous jellyfish that then can get eaten by the stinging jellyfish. So there's a whole food web here that's actually related to jellyfish. And if we look in the middle part of the Patuxent over the past sort of 25 years leading up to about 2003, the volume or the abundance of Nemiopsis, which are the ctenophores, the little blue bulbs on the left, went up substantially. And in the summer, the abundance of the insects of the bay, the macrozooplankton, or the copepods, went down. So presumably those copepods were not grazing on as much of the algae, and the algae went up, right, despite the fact that the nutrients were going down. So it sort of speaks to this just overall cascade in the system that would actually take pressure off the algae and allow them to grow a lot more, just because they're not getting eaten as much. Okay. So I think I want to get to the finish here and skip my last couple of slides. And so I just really want to kind of summarize what I hope you'll take away from this. The first is that the Patuxent estuary, you know, as a whole, has not recovered as fast or as completely as might be expected. But that because the Patuxent is a complex estuary with diverse habitats, maybe no simple conceptual model fits for the whole place. So I tried to articulate that the low salinity upper part of the system did seem to respond to the load reduction, but you actually saw a complete different set of dynamics over time taking place in the lower part. And so thirdly, several impediments to restoration have emerged that would appear to slow the Patuxent's response, but we're not in a place right now where any one of those impediments could we sort of clearly identify as slowing the recovery on its own. And as with all work that we're doing, there's just so many questions that we still have to try to understand the system. We only have a limited amount of data that we can use to sort of have a window back into time to understand what happened. And so the challenge persists for us to consider how we might make new measurements or consider old ones as a way to try to understand how a system like this changes over time. So thank you for your time. That's all I have. And I'm looking forward to taking your questions.
Well, thank you very much, Jeremy. And the questions have been rolling in, so let me try and ask some of them. So the first question is from Ted Turner, and he asks about whether the Patuxent is unique in the western shore tribs in Maryland, so the Potomac, the Patuxent, the South, the Road, and so on. Or do they all respond the same way? And do they all not benefit as much from the saltwater circulatory cleansing that seems to be more present in the shorter eastern shore rivers?
Oh, that's a great question. I have a whole different talk that I can give to try to get at those details. It's hard to say, and forgive me for being inexact, but I would say it's kind of hard for me to come up with a very clear response to that. What I can say is in the back river, in parts of the upper Potomac, in a place called Mattawoman Creek, which is on the Potomac, where there's been really big wastewater treatment reductions, those places have actually shown fairly consistent recoveries. So the Patuxent does seem to be somewhat unique in that it doesn't have as clear of a response to the wastewater treatment loads. But, and there's other features of the system that I think we don't quite understand very well. So for example, for those of you who live on the Patuxent, there's a point just north of Solomon's called Point Patience. And the channel narrows substantially. It's called Point Patience because the current velocities are fairly high as that water gets constricted through the channel. And if you look at the way the water layers itself, it gets really complicated there and far less vertically separated than much of the rest of the river. And it's possible that that's kind of a unique circulation feature. And it could be that it has some impact on how that salt flushing can happen in this system. Maybe I'll leave it at that. I'll sort of log your question and be happy to try to, take a stab at it after, so I can take a couple more.
All right. So several questioners, including Kim Mosingo, I hope I'm pronouncing your surname correctly, and again, Ted Turner, noted data gaps that you commented on in your presentation or that were apparent in your slides. Some about certain parts of the lower Patuxent have not been measured, and some where time series seem to end in 2003. And there are some questions about the reasons for these apparent data gaps.
Yeah, good observation. Some of those gaps exist because I just haven't been able to gather the data sufficiently to present them. But a lot of the ones that are there result because the monitoring programs that were responsible for collecting those data just lost their funding. So for example, the phytoplankton monitoring ended around 2002, 2003. And so we just don't know what happened to the jellyfish and the copepods after that time. The sediment releases of nutrients that you saw, the program that supported that ended around the same time. And until we went out there the last couple of summers on these Patuxent research cruises, we didn't know what had happened in the interim in that time frame. One data gap that I would like to just point out is when I showed the recovery of the grasses in the upper bay, it sort of seemed like it came up and stayed there. I was just looking at the newer data today, and that peak sort of persisted into the early 2000s and then actually started to come back down again. So there are some gaps there. Most of them are due to gaps in the monitoring program, and it's just because those programs are really who's there reliably collecting data for decades, and that's what we have access to.
A question from Sally Homer. Do you think that the loss of oysters might have affected the algal species and density in the Patuxent?
I don't know if I can say anything definitively on that. I will say that the potential for the current levels of oysters that are existing in the vast majority of bay waters are probably not dense enough, or the reefs are not large enough to have a really substantial impact on water clarity or algal biomass. Just to give you an example, Laura Harris, my colleague, and I are working with some folks at NOAA who are trying very hard to document how the restored oyster reef in Harris Creek in the Choptank River is impacting the sort of clarity of the water there. And they think they see a signal, and it might be there, but it's really hard to see. And this is a really small creek that has really healthy reefs right now. My guess, the short answer, is probably not. The oysters that are there right now are probably not having that much control.
Okay, thank you. Another question Is there a way that the public can help collect Patuxent River data to support the research?
I think there definitely is. There are a number of what are called citizen science programs that are operating around the region. And, there's measurements that can be made that are relatively straightforward, like dropping a Secchi disc into the water, or collecting a sample that then can come to the lab. But with proper training, there is an opportunity for citizens to be taking, measurements that can help support us. Learning to use some of the sensor systems we have, collecting water samples and storing them in a way that they can be looked at later. Those opportunities are there. It takes work, and it takes coordination, and it takes a champion. But if it's something you're interested in, I think it's worth getting in touch with myself or Dr. Miller, and we can help guide you to maybe an entity that could help facilitate that.
And just to point out, Jeremy, that there are people who are already helping by providing access to the river through their land, and that in itself is extremely helpful. I know you and Laura Harris were paddling less miles this summer in your kayaks than you would have done otherwise because of the generosity of waterfront property owners allowing you access to the water through their docks, and we greatly appreciate that.
No question.
What is a very simple question, but perhaps not well, in fact, I think there is a simple answer, but I'll leave it to you to answer. Are there parts of the river where fishing and or swimming discouraged?
To my knowledge, I don't think there are any places that I would feel that way. The one thing I think we typically advise, which is based upon our health department colleagues, is that it's not suggested to swim within 24 hours of a relatively large rain event, and that's for the possibility that coliforms can be present in the water. Clearly, I think most of you who follow the news have seen that there are potential infections with Vibrio that happen in bay waters. And, to my knowledge, that's an issue that's pretty rare, but there's just an encouragement that especially in the summer, that if you have an open wound, you just do your best to keep it clean after you leave the water and you're not in any real danger. I fish everywhere in the river, and I touch the water everywhere in the river, and I do it without fear.
All right, and one last question, and we'll allow everybody to go. And it's, you mentioned that water stays around for about 70 days. Have you tried measurements that follow the water over time?
I myself haven't, and those kind of studies are probably the most informative and direct way that we can understand how the water moves. They're pretty substantial experiments, and they're pretty expensive experiments. Given the right numerical model of the system, there are ways that you can effectively simulate a dye getting moved around in the system, and it can tell you that type of information. The 70-day number that I cited came from a really simple sort of coarse model like that. And so there's uncertainty in that number, but it's not inconsistent with numbers that we sort of see for that have been predicted by more complicated models. So.
All right. Well, Jeremy, thank you very much for your time tonight. I want to thank all of the audience members for their participation. I will remind you all that we post the videos of these presentations and typically the presentations themselves on our website. If you go to www.umces.edu and search for Virtual Science Semester, you will get access to all of the information from the talks for this semester and for our previous historical talks. You can see earlier talks that Jeremy had given, earlier talks that his colleagues Walter Boynton and Laura Harris have given. They are all there for your enjoyment. Some of the video quality leaves a little to be desired, but I can assure you the quality of the science in the presentation leaves nothing to be desired. And I want to thank Jeremy for his presentation tonight. I'm going to remind you that we have one last presentation in this series, which is next Tuesday. The topic is plastic in the water and some of the programs that CBL has been involved in trying to reduce plastic into our local waters. We encourage you to join us for that. And once again, please thank Jeremy. And Jeremy, I can only say as a personal note that Michael would have been proud. So thank you very much. And to everybody, please enjoy the rest of your evening. Thank you all.
Thanks for joining. Have a good night.